Okay, picking up with the next lecture video. Bam! So, we were talking just when that previous lecture video ended how this process of cellular respiration occurs in three basic phases. Phase 1, glycolysis. Phase 2, pyruvate oxidation and the Krebs cycle. And Phase 3, oxidative phosphorylation in the electron transport chain. So, details. I know you are craving the details. So, glycolysis. It occurs in the cytoplasm of the cell. So, even though we talk about ATP being made in the mitochondria, the very first phase, the first step in cellular respiration does not occur in the mitochondria. So, glycolysis is anaerobic. What does that mean? Well, let's think for a moment. What is aerobic exercise? You're moving around a lot, getting your heart rate pumped up. When your heart rate pumps up, you start sounding like this. That's me getting up from the computer and going to grab a donut. Aerobic means using oxygen. Therefore, anaerobic is the opposite. Glycolysis does not require oxygen. Important to remember for later in the lecture. So, glycolysis occurs in the cytoplasm and it is anaerobic. In glycolysis, glucose is oxidized to produce 2-pyruvate. So, one molecule of glucose produces 2 molecules of pyruvate. Now, this is not a free or completely spontaneous reaction. It actually requires an energy investment. The cell has to use ATP in order to start glycolysis. Luckily, we get a decent return on investment. Actually, kind of a crappy return on investment. But the point is, you're in the black by the time it's over. So, the costs are 2 ATP in order to start oxidation of glucose. The end products of glycolysis are 4 ATP, and then remember how we have those electron carriers, NAD? Well, 2 NAD swoop in and snag a hydrogen and a spare hydrogen ion. So we get 2 NADH and H. So we get 4 ATP. So what are the net results? Well, the word net, when we talk about the net results, is implying that we take our full production and subtract the costs. So, our net production of energy in glycolysis is 2 ATP. 4 minus 2. So, our net energy production is 2 ATP and 2 NADH and 2 H ions. There it is. Here's glucose. NAD swooping in to pick up hydrogens, 4 of them. And then we also end up with 2 pyruvate as a byproduct here. And here's some ADP plus P, adenosine diphosphate, and a loose phosphate coming in and being converted into ATP, adenosine triphosphate. So, some details on glycolysis. The electron carriers, now that they've been reduced to NADH plus 2H, are sent to the electron transport chain. And then the pyruvates are sent to phase 2, which was pyruvate oxidation and the Krebs cycle. The Krebs cycle. So pyruvate is C3H3O3, reflecting the fact that we've lost some hydrogen ions. And the ATP is produced by a process called substrate level phosphorylation. So, ADP plus P being converted into ATP is produced via a process called substrate level phosphorylation. So what is that exactly? Well, when we have the cell producing chemical energy in order to gain the energy required for reactions, it takes ATP and cleaves off one of the phosphates. So you're left with adenosine attached to two phosphates, adenosine diphosphate, and then this spare phosphate that was cleaved off in order to release the energy held in those unstable bonds of ATP. Substrate level phosphorylation adds the phosphate directly to the ADP in order to create ATP. So again, starting our glucose breakdown requires us to invest 2 ATP. We lose 2 ATP to start glycolysis, but we get back 4. So when we take 4 minus the 2 we lost to start the whole thing, our net production is 2 ATP. All right, into phase 1. That was relatively painless, right? Not so bad. All right, phase 2, pyruvate oxidation. So remember, in phase 2, it's pyruvate oxidation and the Krebs cycle. So, first part of phase 2, pyruvate oxidation. In this case, the 2 pyruvates are converted into a compound called acetyl CoA. So we take 2 pyruvates and convert them into 2 acetyl CoA. What is acetyl CoA? Well, C2H2O is an acetyl group. Coenzyme A binds to the acetyl group. Coenzyme A, CoA, acetyl CoA. Pretty easy. So, the 2 acetyl CoA then enter the Krebs cycle. And the Krebs cycle is taking place in the mitochondria. The Krebs cycle requires mitochondria. Well, unless you're a prokaryote and you don't have mitochondria, in which case all this stuff is sort of occurring at the membrane. Anyway, we're sort of going all willy-nilly and ignoring prokaryotes, though. So, another product of pyruvate oxidation is an additional 2 NADH and 2 H, and those are sent to the electron transport chain, which I'm foreshadowing again. The byproduct of pyruvate oxidation is, well, let's see. We started with C3H3O3. C3H3O3. So what did we lose? We lost a hydrogen, and that hydrogen was, it was sent to our electron carrier, NADH. We also lost a carbon, and we lost 2 oxygen, a C and O2, a C and O2, a C and O2, CO2. Our byproduct is 2 carbon dioxide. So here we go. This is our C3H3O3. We kick off CO2. We get an NADH. Coenzyme A swoops in and binds, and we have our acetyl CoA. And since we start off with 2 pyruvate, well, this happens twice, and we get 2 acetyl CoA. All right, so the second part of phase 2 is the Krebs cycle. Yes, you can call it citric acid cycle if you want, because you're being a jerk and disrespecting my lessons. Respect my authority! Okay. There are 8 full steps to the Krebs cycle. It's kind of complex, but with me along, we're going to go ahead and break them down. And that way, it's not going to get overwhelming. So, you're still going to need to know all 8 steps, but when you think about it, 8, not ton in the scheme of things, so we'll get through it. So let's go slide by slide. One slide per step. So this is the 8 steps here. 1, 2, 3, 4, 5, 6, 7, 8. And we need to talk about what's happening in each step. So with step 1, we have oxaloacetate, and we need to convert it into citrate. So step 1 is oxaloacetate reduction. How does that work? I don't care. In fact, I don't need you to know all 8 steps. What I need you to know is this is where acetyl CoA is transferred next, and I want you to know all the products and byproducts of the Krebs cycle. But I don't care whether or not you know all 8 steps, because I'm an awesome teacher. So, let's look at those products and byproducts. When acetyl CoA is hucked into the Krebs cycle, it goes through a full cycle, and we get a number of products. I have conveniently for you taken into account the fact that we start with 2 acetyl CoA. Remember, 1 glucose makes 2 pyruvate, and 2 pyruvate make 2 acetyl CoA. So for every 2 acetyl CoA, we get 6 NADH. We get 2 FADH2. FAD2 plus is yet another electron carrier, but we only see it during the Krebs cycle. So, yes, you need to know that that's one of the products of the Krebs cycle, but I didn't give you a whole spiel about it during my talk on electron carriers, because really NAD is the primary electron carrier. We're up to something like 10 NADH now. So, 6 NADH, 2 FADH2, what else? 2 ATP cranked out by the Krebs cycle. So, now we are at a total of 4 ATP. 2 net ATP from glycolysis, 2 ATP from the Krebs cycle. Not too shabby. And then the byproducts of the Krebs cycle are 4 carbon dioxide. All right, so my electron carriers, NADH and FADH2, go to the electron transport chain. So here we go. I've circled in red the products and byproducts. This picture is for a single acetyl CoA. So for every glucose, we got to multiply the products here by 2. So we get NADH plus H, CO2, NADH plus H. There's 1 ATP, there's 1 FADH2, and there's an NADH plus H. So 1, 2, 3 for a single acetyl CoA. Since we got 2 of them, that's 6. 1, 2 carbon dioxide for a single acetyl CoA. Since we got 2 of them, that's 4. And then 1 FADH2 and 1 ATP for a single acetyl CoA. We got 2 of them, so that's 2 each. All right. I'm pretty sure my 10 minutes are long up, so we'll pick up with phase 3, the electron transport chain.